Welcome you all to this Cryo-EM course. Last class we discussed about the protein purification. Mostly we discuss about two different types of protein purification. One is affinity purification and another one is ion exchange chromatography. So, now we have to discuss the last purification step. Here you can consider that this is the affinity purification and we ion exchange. So, when we have both ion exchange and affinity purification, what we can do, we can take those samples, this one or two sample, either we do this or we do ion exchange purification. Then one of this sample will take it and will run ACC purification or gel filtration. So, how this ACC purification is working? If we just consider, I'll just start from this side. We have a column, and again this column, it has a different size, and based on that our requirement and how pure we want the purification, based on that we can select that column. It may be preparative column or it may be analytical column. So, what we have some kind of beads inside that column. When we have those beads, you can tell that okay, within that beads there are some empty space. Those empty spaces called the void volume. So, if I just draw all the bits, it will be easier for us. So, bits will pack properly, and now if we just consider that here, if we just look at this one, there is a lot of empty space. Those spaces are called the void volume. So, those void volumes, if we have a mixture of protein, that okay, we have some protein like this, and then we have some protein is like this. So, these are different types of protein of mixture. In one solution we have it, and then we have another protein of mixture which is little bit bigger like that. Okay. So, now this is the mixture of protein, these three component. Now, when we have this mixture of protein, we just pour or inject that entire thing. There will be a valve, and through that valve a pipe will go through this and will inject that protein of mixture. Now, whenever we have a very large size of the protein molecule, so something like those protein molecules, they cannot enter through these beads. What will happen? Those big, they just travel through this void volume, and then they will just avoid those beads, and somehow they will just use that path, and then they will just escape. So, here the large protein, so this brown color large protein, it will just go through that void space, not go inside, and they will elute first. So, when you will use that gel filtration or ACC, that is the reason it is called size exclusion chromatography. So, larger size molecule, they cannot enter to the beads, they will just use that void space and then they will come out. So, when you will collect that sample, when you will collect that sample, first sample, so this is our sample collector. So, there is a fraction collector or sample collector, we are collecting all this sample. So, if we just go here, we can see that the first sample, so there will be a 3 sample collector, I have that I am collecting those samples. So, here based on that size we can get our protein sample or whatever that DNA protein complex, as I mentioned that sometime that antigen antibody complex, that protein protein complex it is forming, and if we just consider that okay only antibody is there. So, if I just have only antibody and then antigen. Okay. So, when we have only antibody and antigen, it has a different size. So, antibody you just consider that 150 kilodalton is the size, whereas the antigen if we just consider that any spike protein for SARS-CoV-2 or influenza is 450 kilodalton. So, if 450 kilodalton and 150 kilodalton is form a complex, it will be 600 kilodalton. So, right. So, now if we just compare this 3 size, this complex has the largest size. So, that time if you just mix these 3 protein together, that this is the one protein, second protein and third protein, this protein will elute first. So, in this particular case, in this one I will get this protein where you have a larger size, okay, because it will elute at beginning, and then coming to the next one, that is my red protein, it is comparatively smaller size. When there is a smaller size, what will happen? They will go inside the bead size and then their path length it will increase, and the smaller protein, so something like that blue, it will go more inside those beads, and then definitely path length will be so big, it will elute at the later stage. So, then if we just have this kind of sample collector, first one will get this one, this is my first collector, this is my second collector, I will get this kind of comparatively larger molecules. Then at the final stage I will get my protein. So, now if you just consider that I have a mixture of this antigen, antibody and then mixture. Now, as I mentioned previously, whenever we are trying to form a complex, this is impossible to get a 100% complex all the time. There will be some unbound antigen antibody will be there. So, when there is a complex formation 100 kilodalton, 600 kilodalton, 600 kilodalton will be elute at the first. The second will be antigen, and then third will be our antibody. Okay. So, this is my antigen, this is my antibody. Now, if you just consider that I have a mixture of these 3, I mix all of them, and then I want to collect my complex and the free antigen also, because that free antigen I can use it for any other purposes to just consider that to generate more antibodies. So, then using this I can use a fraction collector and collect these 3 different protein, and if we just consider that when we will try to purify this protein, you will get a profile like that. So, this may be elution volume and this may be the this is the absorbance, and this is your volume, that elution volume you are calculating, or the fraction, different fraction you are collecting. As I mentioned that higher molecular weight will isolate here, then this will be the medium and this is the lowest molecular weight we can isolate. Now, when using this ACC purification based on the size of the molecule, based on the size of the molecules, we can separate them here. So, in one particular volume, if you just consider that generally very large molecule, we can collect something 8 ml or 9 ml and then 12 ml, then 15 or 17 ml. Based on that size, it is going to separate. When it cannot separate, as I told that okay, we have 150 kilodalton protein and we have 450 kilodalton protein. There is a lot of difference between the size of these two. Now, we have a 650 kilodalton protein. You just consider that I have another antibody, it has some 160 kilodalton. I mix with this same antigen. So, this is my antibody, the another antibody which is coming 160, and then both of them together it will form a complex like this one. Sometimes this is bit difficult to separate this kind of complex. It will be very easy when there is a good size difference, or you have to use a very good analytical column where this 10 kilodalton difference can also distinguish and it can separate it out. Now, one more part that is very important here. So, most of the time as I draw that SEC profile, right, I draw a SEC profile, you just consider that you have a SEC profile, something like that. And similar time you have another profile, okay. So, there is a two different profile, it is coming at the same position and this one is the last one that okay. So, I will take a separate one, always remember that absorbs here and this will be the volume. Okay. So, this is another profile that okay, we are getting something a very large profile. So, this kind of profile probably at the peak region you are getting your maximum protein, whatever you are thinking it is your required protein. This kind of protein, when your SEC profile is like this one, it is a very difficult to tell that you have the required protein and you purified your protein properly. So, this is not a very good profile. Most of the time we are expecting a very sharp peak when you purify our protein. So, it will be very sharp. So, you will only consider the protein from the peak area, or you may check that two different solar. There are many people or many researcher I noticed that or students they are doing that they purified the protein and collected all the fractions together. So, then the running the SEC purification is useless, because SEC purification we are trying to separate our protein based on their size. And now, if we are not paying attention in this particular stage, that here in this particular purification you can see there is a peak and there is this is the peak we are getting, but there are lots of other proteins are mixed here in this region. Now, if you try to mix entire thing here, what is happening? There are some larger size protein and there are some smaller size protein, all are coming together. So, this is not any good purification, this one is the mixture of all the proteins. So, better if you have this kind of purification, take the peak area and re-inject or re-purify again. Sometime this broader peak, this red we can tell that this is little bit a broader peak, this red one. Now, if we want to collect all the fraction from this to this, so all the fraction means it is mixture of those large protein also and those small proteins also. So, most of the time we should select only the peak area or the right hand shoulder or the left hand shoulder. If you have a very sharp peak, something like that, if there is a no change, you will just check the peak area for your rest of the experiment, especially for structural biology purposes. If you want to collect a cryo image and a homogeneous sample, most of the time, even if your peak is like that, it will be better to use only that peak area, or check the left hand shoulder and the right hand shoulder and take only one fraction. If you just consider, if this is your F10, so this will be the fraction collector 9, this is the fraction collector 10, this is fraction collector 12, and this is fraction collector 8. So all this fraction it is collecting. Now if you mix all these fractions, means you are adding some larger size, some smaller size, and your required protein. So your sample will be heterogeneous. So that is the reason most of the time we just want to select the peak area for structure determination. Okay. So next go to the slide and take a look what is happening. What I discussed so far, the same thing is discussed here again. So this is the gel bead and those gel beads are here, and this is the matrix, and then whatever the larger protein, as I mentioned that blue is the larger protein and red is the smaller protein. When you have this mixture and the sample is going through that column, what you can see, this blue will not enter through the gel. Here you can see those gel beads, only that small protein and red proteins are going inside this bead, and the larger one is not going inside the bead, it is just going through the void volume and eluted at the beginning. And then whenever this protein, small protein, is going inside the gel beads, they have to travel longer distance, and that is the reason they elute at the later stage, and you will get this kind of profile. So this is the volume of elution, that okay, how you elute your protein. So this will be the larger protein and this will be the smaller protein. Now this is the most important part. Sometime when you purify our protein, as I mentioned that many cases we are using a very high salt concentration or very high detergent concentration, and not detergent, for the membrane protein we are using detergent also. Sometimes we use a very high detergent concentration. The membrane protein sample preparation will when we will discuss about that why detergent is important. Sometimes we use detergent, sometimes we use salt, sometimes we use glycerol, and now we have to remove those salt or glycerol from our sample. So what is the best way to do it? Here, that is called the dialysis, to separate the small or large molecules. So generally there is a dialysis bag, it has some kind of pore, and those pore sizes are very small, it will be in between 1 to 10 nanometer. Now so we have a protein of interest, that is we already discussed that okay, this is my protein of interest, and now very concentrated sample, and we put inside the dialysis bag and then we tie up both the ends, and now there is a buffer solution. In this buffer solution, so if you just consider in this protein of my protein of interest is one of that blue protein or that red protein. Now so if you just consider, now this is this blue is my interest and blue is the large size, and those red dots here basically are the high salt or the glycerol molecule. Now I want to reduce that. So what I am doing in this solution, in this buffer tank, there is no glycerol or no high salt, and we just try to keep this dialysis bag for overnight. Then what will happen, gradually that highly concentrated sample inside that bag will slowly diffuse from this dialysis bag through that nanopore. When that salt or glycerol will diffuse to the buffer, inside that bag majorly it will stay protein, and very small amount that salt concentration or the glycerol concentration. It will keep going on like that, the molecules will go out based on that, based on their until it will reach to the equilibrium. So, this buffer solution and inside the bag, when it will reach to the equilibrium, all this movement it will stop, but this is how we can remove most of the time that high salt or high glycerol concentration inside inside our protein. So, this is very much required. Why it is very much required? Because most of the time it is a very high salt concentration. This granular salt, you can see it when you will image the image using the high resolution TEM, because here we are not using using an optical microscope. Here we are using a high resolution transmission electron microscope, and high resolution transmission electron microscope, it will always give even if it is a high salt concentration, all these salt molecules will be visible when you will do the negative staining, as well as the in cryo images also high salt concentration is going to affect our cryo freezing. It can change the surface property of the grid and it will affect our cryo freezing, and most of the time glycerol is inhibit the contrast of the sample. That is one of the reason we prefer to remove the glycerol or any organic solvent. Now, we are going to the next step. The next step, that okay, we have performed all types of purification. So, all types of purification means we did affinity purification, ion exchange purification. From these two types of purification, we collected that protein sample and then we perform the ACC purification. ACC purification basically based on the size, it is separating. Now, first step is a very crude method, that affinity purification, the crude extract we are just transferring through the column, and then we got a almost pure protein. Then we are performing the ACC, where we are getting a very clear, neat and clean protein. That protein we are taking it and doing that dialysis to remove the high salt or glycerol concentration. One thing we all should remember, that whenever we will remove that glycerol or high salt, if our protein is very happy with this high salt and glycerol, so it will be a little bit tricky to remove those component, because then there is a chances are very high that protein will precipitate or it will aggregate, and when you will do cryo or TEM imaging, we can see this kind of problem. Sometime during the purification we can use high salt concentration and glycerol, and then if we just remove most of this high salt concentration and keep little bit amount of salt, means 100 millimolar or a 0.1% glycerol, protein will be happy, soluble protein, there will be no problem. That is the cases we can do rest of the experiment slowly, but if there is a tendency that after our dialysis, when you will remove the high salt and glycerol concentration, and there is a tendency that if we keep the protein at 4 degree for longer time, protein may precipitate or protein may aggregate. So, better to take that protein immediately and go for TEM study or cryo study, do not wait for long time. So, this is recommended that if you really want to do structural study or any imaging, it will be better to use your fresh protein and then immediately try to image it using TEM, and if sample looks good, immediately you transfer this sample for cryo freezing. In the same day, if it is possible, it will be always good strategy. Now, this is the condition when our sample is almost ready for TEM imaging and cryo freezing. Now, before going to the TEM imaging or cryo freezing, it will be better to do little bit more analysis of our sample, and those analysis will be some biochemical analysis. One of that analysis is this one, that gel electrophoresis. So, this is a polyacrylamide gel electrophoresis. So, this is the apparatus where you have the buffer solution and there will be a charge difference, and your protein, whenever you will get that protein, will take that protein and mix with SDS. SDS is some kind of detergent, and that will give you a negatively charge of your overall protein and also denature the protein, because overall protein charge, if it is negative, all the interactions whatever is responsible for forming a helical structure or beta structure, that will disrupt, and your protein will be in a linear form. And then you will add SDS, also you will add the beta mercaptoethanol to rupture the disulfide bond. So, then linear protein, you load it in this lane, and then there is a negatively charged, and here you have a positive charge. So, and your protein is also negatively charged because sulfate group is attached from the SDS. So, when your protein is negatively charged, and if this charge is going from negative to positive, so protein, if you are loading your protein here, protein will start migrating through this polyacrylamide gel, and based on your size and charge, it will migrate for a certain time and you can get your required protein. If you have a marker protein where you know the molecular weight of that protein, and if you load that protein, so you will get the exact size of that protein. So, here this is a marker protein where we know that size of that protein and how many protein components are here. So, in this marker we have total 6 component. So, in that we have a protein 14 kilodalton, 21.5, 31, 45, 66.2 and 97.4 kilodalton protein. Now, here you can see this is my required protein, where we mention that purified protein, cation exchange protein, anion exchange protein. Using this kind of purification, we isolated those protein, and here this is the soluble extract and this is the uninduced. So, in this particular case you can recognize that this is after induction, your protein is overexpressed, which is not present in your induced cell, uninduced cell. Induced cell you have it here, this is the uninduced cell and this is the induced cell. In induced cell you can see that band. Now, the soluble extract, you have multiple other proteins, and then you have your protein also in a huge amount, because in induced cell you have this band and in soluble fraction also you have this band, but it is missing in your uninduced cell. But if you go in the purification stage, you can see in your soluble extract so many proteins are there, but when you use the anion exchange or cation exchange or purified protein, you can see only single band. That means after your purification you are getting a very pure protein which is matching with this induced cell and the soluble extract. And now, if you consider that molecular weight of this protein, it will be roughly 45 kilodalton. Now, based on your literature and our other experimental study, you know that your protein should come at 45 kilodalton, and from this marker, when you are getting a very clean this kind of band, that will give you this confidence that I have purified my protein and this is my required protein and I can go for further study. But now suddenly you are expecting a 45 kilodalton protein and you are getting a band somewhere here, you are getting a band somewhere in this region. That means this probably is not your protein. So, better you take this protein and confirm it, is it your required protein or not. If it is not your required protein, there is no point to go for any cryo-EM study or TEM study. STEM study is still okay, but for cryo-EM study most of the time it is a very time consuming study. So, that is the reason it will be better to confirm your protein first, that okay, this is my required protein, and then go for the next step. So, I will just go through a flow chart. There it will be clear that what kind of steps we should follow during our experiment. So, this is a very I selected this one from a book chapter by low repass mode. Here they discuss this sample preparation techniques very well in this book chapter. So, where first thing I already discussed multiple times that okay, protein preparation, and that is the reason I spent so much time in this particular step, where this is your SDS page gel, or you can try 90 page gel to confirm the oligomeric stage. So, here you have the marker and the requirement, then you are performing the ACC and you are getting a very sharp peak which is good for rest of the study. So, from this one what you will get to know? You will get to know the composition, purity, homogeneity, stability also, that sometime if you change the buffer composition, that you are changing glycerol, you are changing the salt concentration, in that condition your sample is stable or unstable. If it is unstable, then you cannot remove entire glycerol or salt, you have to optimize those conditions. And then biochemical activity, this is very important. If your protein has an affinity to DNA or RNA, or it has affinity to some kind of ligand like ATP, it will be better to do the activity test, because when you have a recombinant protein and if your protein is not active, even if you go through this entire process and determine the structure, that will be completely useless. So, that is the reason it will be better to confirm that your purified protein is enzymatically active and then go for the next step. What is the next step? The second step, as I mentioned, that will be the negative staining TEM. So, here you can see this is a negative staining TEM. We will discuss these things more, where you can see this is the background and this white molecules is your protein, and this is the black patches, basically that is your staining solution. You should see, whenever you do the negative staining, you should see the discrete particle stability and particle size and shape. So, if you have a ribosome, and if ribosome, these are the ribosomes molecule, and ribosomes if it is looking like a circle, so then definitely something wrong with your purification. So, from your negative staining images, if you have a reference images, it will be much easier for you to compare and figure it out, are you looking at a correct sample or not. When you have a correct sample, you go for the cryo-EM study. Cryo-EM study also same thing, you have to pay attention to the stability, because lot of time due to surface tension or some other forces your protein may disrupt, or it may form a small particles, or it molecules can come together and form a aggregation. So, you should see the isolated particle, they should not be overlapped with each other. From there you can consider the particle size and shape and the particle distribution and the concentration. Both the things are very important for the single particle analysis. Then these are the two steps at the final stage, when you will use this particle to calculate the 2D classification, initial model, structure calculation, and finally we will get that final structure. These two part we will discuss at the end. Recent part we are going to discuss about this negative staining and then cryo-EM study. So, today's class I will stop it here. In the next class we are going to discuss more about that cryo-EM or TEM grid preparation or sample preparation. Thank you all for your attention.